Hello everyone! In this video, we'll be explaining to you the affinity chromatography general concepts and protein AG as an example. We are Ji-Yeon, Farzad, and Salma.
Have you ever thought how to selectively separate your products after the production step of a bioprocess? Well, consider affinity chromatography as a great strategy. So I'm gonna tell you an interesting story about how affinity chromatography got to become so familiar with us these days. Between 1950s and 1960s, there were several original researches describing isolation of enzymes by ligands of selective chromatography steps. One example is the use of flavin cellulose compounds in column chromatography to purify liver flavokinase. In the 1968, the term affinity chromatography was first used in a seminal article written by these two, Pedro Cuatrecasas and Meir Wilczek, who both later got Wolf Prize in Medicine in 1987 for their contribution for invention of affinity chromatography and its applications to biomedical sciences. In the 1970s, various ligands capable of interacting with several targets were introduced, such as protein A, coenzymes, lectins, nucleic acid, and etc. Around the same time, hybridoma technology was also developed to produce monoclonal antibodies, which started the wide use of monoclonal antibodies in biomedical separations.
In affinity chromatography, the general separation principle is based on a highly specific macromolecular binding interaction between the biomolecule and another substance. This interaction can be between an antibody and antigen, or between an enzyme and substrate, or a receptor and a ligand. Different types of binding interaction commonly used in affinity chromatography procedures are summarized in this table. In general, all types of affinity chromatographies are composed of three steps. In the first step, or the loading step, the mixture of different molecules is passed through the column, and the binding partner inside the column captures the molecules with specific affinity. Non-targeted compounds are washed away from the column in a wash step. And finally, a buffer with different ionic strength or pH will disrupt the interaction and release the molecule from its ligand as a purified product. One of the best examples of the affinity chromatography is the protein A or protein G affinity chromatography for purification of monoclonal antibodies. Protein A is a surface protein from the cell wall of the Staphylococcus aureus bacteria, and protein G can be found in some group of Streptococci. Both proteins can bind specifically with strong affinity to the Fc portion of the immunoglobulin molecule. Therefore, considering this unique feature, both proteins can be used as a capturing part for the purification of different antibodies which are produced inside the laboratory.
The uses of affinity chromatography mainly include separations, purifications, analysis, or characterizations. Many of the applications involve both small and large-scale purification of biomolecules, such as enzymes, native and recombinant proteins, by using dye-ligand or biomimetic affinity chromatography. Common examples of popular small-scale applications are isolation of histag proteins by immobilized metal affinity chromatography, and isolation of specific antibodies or antigens by immunoaffinity chromatography. However, this technique could also be used for other applications, such as cell affinity chromatography, to isolate certain types of cells by using interactions between cell surface agents and their ligands. It is also used for analytical methods for different purposes, such as separations or analysis of chemicals in samples, sample pretreatment prior to further analysis, or chiral separations important for drug discovery and development. Also, for the examination of biological interactions to characterize their binding strength, their kinetics, and the number or types of sites that are involved, for example, between proteins or drug protein, enzyme inhibitor, antibody antigen, etc. Finally, it could be used for indirect detection and measurement of a target analyte by chromatographic immunoassays.
The support or matrix in affinity purification is any material to which a biospecific ligand is covalently attached. Typically, the matrix is a solid and called resin. Agarose, cellulose, dextran, polyacrylamide, latex, and controlled pore glass are some of the solid supports for binding of the interested ligand inside the column. There are many different types of resin available in the market, and based on some factors including mechanical and chemical stability, flow characteristic, and the most important one, based on binding capacity, they can cost between few hundred to $1,000. Volume of the sample that can be loaded on a column in affinity chromatography directly depends on the binding capacity of the column, and it varies from millimeter for the yield of few microgram protein in small scale columns to cubic meters of samples in the industrial scales to obtain grams of purified protein. Based on the mentioned factors, there is a wide variety of the column ranging from microliter up to 200 liters for purification of different products.
There are many manufacturers providing protein A and G products for antibody purification, such as Thermo Fisher, BMake, Santa Cruz Biorad, Sigma Aldrich, and Abcam. For example, Thermo Fisher provides different kinds of protein A and G products based on their different properties, like bead or resin size, or binding capacity with different kinds of antibodies. Affinity chromatography is so versatile in downstream process. It can be used in many different stages in the downstream process, including capturing steps and policing steps. So affinity chromatography is mainly used to capture the target molecules from solution of mixtures, as seen in this picture. However, it can also be used in policing steps, such as removal of process-related impurities when used in flow-through mode. The resins in this case are called scavenging affinity resins, or they can be used to separate the desired form of a target molecule from closely related product impurities by, for instance, selective elution. The most common types used in affinity chromatography is protein A and G resins and histidine-tagged proteins, and they show common yields of 50% and 90% each for a single step, which can be lower or higher with different operation conditions.
Now, the purification step in a downstream processing scheme can be also performed with other technologies. For instance, with ion exchange chromatography, where the separation is based on electrostatic affinities. This method can sometimes be a good alternative to purify monoclonal antibodies of a basic isoelectric point, because the cost is almost one-fifth compared to protein A chromatography. Another alternative format is membrane chromatography, which combines the advantage of membrane filtration with liquid chromatography and is cost-effective to purify a large volume of feed streams. Many innovations have been recently attempted here, such increasing the binding capacity with hydrogels and nanofiber-based matrices. Another alternative format is annular continuous chromatography, where the chromatography bed is packed into a rotating annulus to work in a continuous mode. It has been used to purify recombinant blood clotting factor IX, virus vaccines, and recombinant antibodies from the cell culture supernatant. Finally, there is multi-column countercurrent chromatography, where at least 3 columns are required. The first one is loaded to saturation. The breakthrough is loaded onto the next columns, and the saturated column is washed and diluted as in a batch operation. This one is useful for resolving various protein variants with high yield and purity of the target product. This is the case of commercially available recombinant streptokinase, single-chain antibody fragments, and various monoclonal antibodies. The first advantage of affinity chromatography is that it reduces the number of purification steps, which thus increases the overall process yield and reduces processing time. In this figure, we can see the comparison of non-affinity and affinity-based purification processes. Even if the two first non-affinity steps have much higher loading capacities, using affinity chromatography as the first step can reduce the overall cost of goods 1.5-fold as compared to the non-affinity process. This is because the first non-affinity step has a much lower yield, as we can see here in the graph. Thus, the overall process yield ends up being significantly lower. Also, costs can be further reduced by increasing resin cycling and improving target loading. For instance, protein A resins are now routinely cycled between 100 to 200 times. Now, affinity chromatography also allows for standardization in different cases, for example, through platformization. This refers to a highly selective affinity chromatography-based capture step, followed by a few polishing steps. Typically, each company develops a few variants of a platform process with different unit operations post the capture step, in order to properly purify variants of a target molecule. So in addition to standardizing manufacturing operations, this concept plays a vital role in scale-up and technology transfer, as well as in defining layouts of manufacturing facilities. It also shortens timelines and reduces cost of process development, because a very robust downstream purification process can be developed and scale up with a lower number of full-time equivalents. Finally, this standardization also enables a continuous and intensified mode of operation. Now, the particular advantages of protein A affinity chromatography include its simplicity and large purification factor, but also the specificity, because it relies on a very specific interaction that is not sensitive to variations in the feed stream composition in comparison to other techniques. Also, because of its high conformational stability, protein A is able to refold after denaturation, it resists cleaning with reducing agents, it has a fair degree of steric mobility, and its binding domains are protease resistant. Finally, this step is great for volume reduction since it provides a 5 to 10-fold concentration of the product.
Despite all the benefits of affinity chromatography, there are some limitations too. First one, it takes a lot of skills to handle it. Affinity chromatography requires some good technical skills to perform effectively, and sometimes it can be tedious and time-consuming, especially in the case of large-scale samples. The second limitation is related to the volume of the sample, which depends on the binding capacity of the column. Third one, the ligands used in affinity chromatography are costly. Overall, the demands for specific ligands make this process an expensive technique. Sometimes, also, the ligand leakage can be observed from the affinity columns, and it is directly affect the purity of the product. Also, there are some non-specific adsorption can be observed in the case of the some affinity chromatography techniques. And finally, using the extremely low or high pH solution and buffers will be resulted in degradation of the solid supports and limited lifetime of the resin.